right, so I'll go ahead and introduce. So I'm Rich Lenski, for those of you who don't know me, and it's my great pleasure to introduce Dr. Betül Kaçar. I met Betül at a Gordon conference some years ago. I forget exactly which year it was, but after talking to her there and seeing her at other meetings and following her online outreach activities and reading her research papers, I'm just a huge fan of Betül's work. I think of it as kind of a mixture of synthetic biology meeting experimental evolution with sort of a biochemistry and ecology twist to it all. So she does things like reconstruct ancient proteins based on sort of using phylogenetic inference to understand what the sequence of those genes was. And then she engineers them, puts them into modern cells, and looks at how they evolve in one set of projects. In other sets of projects, she does things like look at how those genes are expressed in modern and in reconstructed ancestral or paleontological conditions, say pre-oxygenated environments. But besides her fantastic research, which you're going to hear about, she's just got a huge focus on science education and outreach that has an impact related not only to evolution, but the field of astrobiology. And she does it not only here in the United States, but she does it around the world. Betül received her undergrad degree in chemistry from Marmara University in Istanbul. She then came to the US and did a PhD in biomolecular chemistry at Emory. She then did a postdoc at Georgia Tech, and then she got a NASA postdoc and set up and led a research group at Harvard. Then in, I think, 2017, she joined the faculty at the University of Arizona, where she has joint appointments in the Department of Molecular and Cellular Biology on the one hand, and the Department of Astronomy on the other. But she just announced that she's accepted a position and will be moving in just a couple of weeks to the University of Wisconsin, where she'll join the Department of Bacteriology, a department with a really fantastic history and faculty. And so we welcome her to the Midwest as well. Betül has won lots of honors and awards, and I'll just mention a few to sort of show the scope of her impact. So she won a HHMI Curriculum Development Fellowship at Emory. Then she had this NASA postdoctoral fellowship. One of her cool honors was she won the Way Cool Scientist Award from the Science Club for Girls. She's given a special lecture on astrobiology to the Library of Congress. She's been a participant in something called the Science Exchange Program, sponsored by the National Academy of Sciences, which brings together young thought leaders in various scientific areas. And so it's just a tremendous pleasure for me to welcome Betül, and she will give her keynote talk today on exploring life's early evolution using ancient enzymes as paleosensors. So take it away.
Thank you so much for having me. This is very special. Beacon and members, of course, have been a great source of feedback for me for years. I really think that the feedback and the comments and the questions I received from this very community is the foundation that shaped our work to be where it is today. So it really means a lot that I am here talking to you today. So we, as Rich said, we combine everything, and I like to call it as it's not a fruit salad, but it's a smoothie, and then we try to apply it into questions that hopefully will reveal important impacts about early life and origins, with application hopefully in the decades to come to our search for life in the universe. So I am also foundationally interested in understanding this dynamics between chance and evolution, and I know the majority of us here are. But we want to give this a spin and try to understand whether we can address this at the planetary scale, and see whether. I'm so sorry. I will have to move this here. Right. So we want to understand at the foundational level what are the planetary environments, and how are the planetary environments may affect the biological life, its direction, as well as, of course, its emergence elsewhere. And so this is really why our work is, I think, rooted for applications for NASA, given the fact that we are finding, not only finding, but also will be visiting different worlds in years to come. They really are interested in investing in biology, biological sciences, to address this question from an evolutionary and molecular view, which doesn't exist right now. We have tremendous amount of information coming from geology and paleontology, but really a foundational understanding of the molecular roots is lacking. So we've been developing systems, and as Rich mentioned them, by combining phylogenetics and experimental evolution. And it's been really a journey towards, finding these systems and developing them in a way that they will be the most informative in our understanding of the past. That how do we explore other possibilities of life as we know? Can we leverage synthetic biology and designing new enzyme systems, designing new genetic systems using whole organisms as a shuttle, so to speak, in order to target early life questions related to early life. The key, I think, really is that comes from integrating these two seemingly unrelated, but of course aren't, records of life. One of record that we really understand when it comes to the past comes foundationally from geology. We have fossils, we have geochemical records, we use these different classes of fossils and chemical records to understand the past environments. Of course, we are limited in what we can find, right? So these fossils need to be conserved, and they need dating them can be hard, and ultimately we are trying to read some rock in order to ultimately understand the microbe that once lived on this in this locality. And then from that, whatever we find, we make this huge inferences about how life in the past might have been. It's quite tremendous, actually, what we are able to learn from this. And of course, on the other hand, we have the molecular record that really relies on extant molecular diversity. And we use molecular clocks or phylogenetics in order to make sense of what we see around us today, and then tracking our way back into the past using multiple tools. But of course, the trick with the biological record is that it's overwritten. DNA is an ever-evolving molecule, unlike rocks. So good luck, right? Trying to understand something 3 billion years ago using current DNA as a system. But really at the heart of these two different records are enzymes. I love enzymes, right? So they're really amazing. Yes, maybe they're small and tiny, but they do amazing things at the global scale. So we know that they catalyze many amazing biogeochemical reactions. They are the drivers. They are really essential partners for many important significant metabolisms. So today I want to talk to you about nitrogen fixation in particular, and an enzyme that plays an important role in nitrogen fixation, which is nitrogenase. And I want to talk to you about why we chose nitrogenase, what was our journey towards that. And some of you may realize how the questions you asked were received by our end, that we really integrated them in designing these experimental systems. So really then we see enzymes as the mediators of the two records that have their own strength and limitations. So the question is then, how do we integrate the evolution of enzymes with the geologic record to then perhaps not necessarily maybe fill in the gaps? Of course, we would be happy if we do that, but complement this data, maybe even contrast it. So that was really the starting point for us. So what do I mean by enzymes playing an important role for the rock record? As I said, if you're lucky, you'll find a fossil, and then maybe you'll have some result, some value that is leading to a promising biogeochemical biosignature, a signature of some organism that was left on this location. But really, if you dig deep, what you're really doing is that you're trying to understand how microbes selectively use the different isotopes of certain elements in the atmosphere, and then chewed them and digested them and spit them out in distinct ways that allows you, hopefully, to differentiate what is biological and what is not. And that's a very big problem, of course, to understand whether you're looking at a biotic record or a biological one. And the only proxy we have right now is modern life. So the values that we characterize from these different isotopic records, and then by the distinct ratios and that we obtain from preserved organic matter, are ultimately compared to the modern taxa. And then by understanding the modern microbes, their locations. And this is, of course, also ever changing right now. It's an amazing time to, more than me, better than me, perhaps, that how these amazing microbes live in very different environments. So we then compare these ancient records with the modern ones and then try to understand their location. This is exactly what we do on Earth. This is what Curiosity does on Mars. This is what we will do when we visit different planets. This is really a foundational measurement of understanding biotic life from and differentiating it from a biological one. So we started with this, okay, we have this record, we compare it to the modern proxies, but are there alternatives? How do we really know that the past was the same? We are really taking this for face value and assuming that the past life is similar to the modern life. Of course, we need to make this assumption because we don't have a time machine. We can't go back and study these things in situ. So we need to compare them to the modern versions, but we don't quite, I think, make a lot of assumptions here when we make these connections. And this is where I think alternative systems, synthetic biology, creating these sort of evolutionary synthetic systems may be useful so that we can try how can we modify these things and analyze them. And that's what I want to talk to you about today, that how we create this synthesis. So we refer to this as a molecular paleobiology approach. And this is really the smoothie or attempt to make a smoothie. And of course, one thing about smoothies that you have to be careful. If you have a rotten fruit in there, the entire thing is bad. Okay, you have to be very careful with your ingredients. It's a similar thing when you form a team. You got to be very careful with what goes in there, because once you mix them, there's no going back. So in our case, of course, we did a lot of alterations, but we usually start with phylogenetics, create trees, and then reconstruct different proteins using our phylogenies, and we target different ages. We ultimately engineer them inside organisms to see how they will impact the behavior of the organism, to then ultimately connect it to the rock record. So this final bit, as I said, I think really connected what we were trying to do over years. We started this work with translation proteins and then inserted different initiation factors, elongation factors inside E. coli. But you can imagine that there are many ways to break a translation machinery that may have nothing to do with the historical constraints. So we moved on to metabolisms that actually are historically distinct and leave a remnant in the rock record. Of course, we are continuing our translation work at the same time to answer different questions. But going back to the system, we then switched the gears and looked for enzyme systems that at the end are functioning at the very basic reduced molecular level, but are able to impact the organism and an ecosystem, even at the planetary scale, what an entire planet may be able to give away as a color or as a distinct a shift in the atmospheric signature. So how do we do this? So this is the sort of the framework that I was working on when I was a postdoc, and so we decided that you have to check different points if you want to create a system like this. First of all, you need an enzyme or a different, also a geochemical record that is related to a protein function. So you cannot just, again, translation machinery doesn't have a direct geologic history, but nitrogenase or Rubisco are. Right, so there are different metabolisms that are directly related to the functions of these enzymes. Knowingly or unknowingly, we make these direct attributes. Yet again, I don't think we dig in enough to see if that's actually the case. And of course, we need a phenotype. Are you gonna create a sort of a fitness value, or what kind of behavior are you trying to analyze here in order to make this connection? Is it an isotopic discrimination value? Is it just a growth value? How will you connect what you see to the past? And of course, the organism you're engineering has an ancestry. If you don't want to engineer a spinach if you're interested in deep time, but you may want to go for cyanobacteria because it's really old, prior to the as old as probably over 3 billion years. So with this paradigm, we then hit the road that not only we reconstruct the ancestral genes using phylogeny and making inferences using focusing on distinct nodes along the trees, but also connecting them to ancient biogeochemistry by treating them as though they are sensing an ancient environment directly. And we are doing this right now by doing environmental interpretations, by measuring their isotopic outputs, by measuring these engineered organisms' isotopic outputs. So we're very excited about this. It took many years together, and so it's pretty fine. So right, this is the system. So let's now take a step back. Why is nitrogen fixation interesting? What is the real big problem that we're trying to focus here? And you may also know that we actually just got a NASA center. This is a multi-million-dollar research center. I'm honored that I will be directing this. And this is the very starting point for the center over the next 5 to 6 years. So we want to understand, and then now that we have this system, can we apply this to understand the foundational questions also related for astrobiology, and also I think most of us, that how do we discern and understand the dynamics between the environmental availability of a certain metal or an element versus the functional properties that it triggers in biological systems, and how the biological system may respond to this distinct element. So we want to understand this dynamic, that just because a certain element may be present in the environment, does that mean that biology and life will select this environment just because it was around? Or are there more evolutionary constraints that impacts the selection of these enzymes that may not have anything to do with the environmental abundance? I think we again write these just those stories in our minds that, oh, life chose iron because there was a lot of iron. So that seems to be the assumption that we make, but we wanted to challenge this. Could this be right? Especially when we know that evolution does tricks that we cannot even imagine. Experimental evolution showed this many, many times. And we don't quite understand ancient environments. So should we just assume that just because something is available in the environment, that's the reason why we see the innovations that we see in the past? And this is also, of course, important for today's life. We know that the elemental abundances determine the distribution of life on Earth. This is a satellite image from NASA. I think it's not the newest one, but it still nevertheless shows that where the biologically productive areas are in the globe, and how they may overlap with the different abundances of different elements. And how this dependencies evolved was really the starting point for us. This is a very important question for Earth system science, but again, lacking is a molecular and mechanistic view, I think, when it comes to understanding this selection. And that's where the sort of the beacon thinking shines in for questions like this. I think that really understanding the mechanism. So we named this "What Does Life Want?" We want to ask this question straight head on. That let's explore the natural selection of chemical elements and understand, is it a function or an availability issue, or is it just a historical accident? How can we explore these questions? So our proposal actually does include a lot of experimental evolution. So I'm hoping in the future to be able to share results from that. We'll be setting up chemostats and evolving different systems. So we can talk about that hopefully in years to come. But so let's look at the metal abundance versus biology. What do we so far know? And again, primarily, maybe dominantly, really, this comes from our knowledge comes from geology. So we're looking at the history of life on Earth. Here we are present, and this is the past. These are the 4 main geologic domains. So I'm not interested in Phanerozoic or Proterozoic. Okay, life happens, so many things. So let's go back, right? Let's go back to the roots. And here is the different oxygen levels on the y-axis. We know that early Earth did not have oxygen, and then we have this remarkable innovation with the great oxidation event that we have oxygen on the planet. Of course, it's a big debate of how did this happen. But we also know that the metal availabilities did not stay constant over geologic time. So we know that pretty much that iron, manganese, or cobalt were pretty high amounts in early Earth. And then the amounts of these metals varied over time. For example, molybdenum wasn't in high amounts in ancient oceans. However, when you move to for about 2.5 billion years, molybdenum amount increases while the iron or zinc amount decreases. So the elemental abundances over time shift. And looking now, we have pretty high molybdenum, I think in fact highest, and not showing here in vanadium amounts are also variable over time. So we have some idea about what happened in terms of the elemental abundances. But of course, the related question is that what metals did early life use? So here's by the way, a straight example of how, of course, environment in the past wasn't the same, and environment directly influences life one way or another. So here we go. But which metals then did early life use, and were those choices driven by the availability of these metals, or what sort of evolutionary forces drove them? So we turned into nitrogen fixation for this problem. It's an excellent system for many reasons. First of all, we don't know much about it. We don't know much about its origins and evolution, so that makes it great. It's also a party of all the metals, really. They're different. The molybdenum, vanadium, iron, they all get together to enable the catalysis of a protein that is functioning at the heart of nitrogen fixation, that is nitrogenase. So not only it's an ancient system, I'm able to talk to you right now, probably because of this innovation. We are here based on the fact that nitrogen fixation is enabled fixing the atmospheric nitrogen and turning out, through the catalysis of nitrogenase, turning into ammonia. This is the only way life does it. And an enzyme at the core of this enables this catalysis. So we focused on nitrogenase, not only because it is a critical enzyme for nitrogen fixation, but when it also comes to nitrogen isotopes that we use in order to interpret the age and the presence and the different function of nitrogen fixation in the past. This enzyme is the main, is the rate determining, catalyzes the rate determining step. So this is also an enzyme that has a direct connection to biogeochemistry that I was mentioning in the beginning. Amanda Garcia, Dr. Amanda Garcia, incredible postdoc, really came to the lab head on with this. She was trained in paleontology, so she learned how to clone, how to do experiments in the lab, and was an excellent candidate for this work. And what I will present are her work, her results directly. So again, we don't know when nitrogen fixation evolved. We have some rough idea. We are looking at our timeline again. Origin of the nitrogenase and nitrogen fixing enzymes is roughly, and this is a big rough, between 3 billion years to 2. So we think it's somewhere around here. But we don't know exactly what sort of components were present back then. Was it the nitrogenase as we know? Was it a different system? But we set course to not only reconstruct nitrogenase, but also study the ancestral enzymes, but also engineer these ancient nitrogenases inside nitrogen-fixing the prime nitrogen-fixing bacteria, azotobacteria, and then see how this ancestral system will impact the way bug is uptaking different the nitrogen amount in the atmosphere, but also different isotopes. So we developed a system for engineering, but as well as assaying. I'm going to show very, very preliminary results when it comes to the actual experimental data, but I'm going to run you through what we've done to understand this ancestral enzyme first. So we have and I just want to check the time. Are we good? Is it like 5? Okay. So right, here we are. So nitrogenase. We have the FeMo-Co. It's a iron molybdenum cofactor in the heart of this enzyme. And molybdenum preferring nitrogenases are the prime nitrogenases today. So majority of the organisms that fix nitrogen and have nitrogenase does that through a molybdenum preferring enzyme. So that was an interesting enigma for us to start with, especially, and the careful ones in the audience will remember this, that the molybdenum amount in early Earth was low. So tada, like this was great. Right? We have an enzyme that prefers molybdenum, but it evolved in an environment with low molybdenum. So what did the ancient version have? So this was our starting point. As I mentioned, this is a metal party really with the molybdenum, vanadium, iron, nitrogenases, all preferring different, in different nitrogenases. These are not very easy enzymes to study biochemically. There are a few leading labs that have these amazing systems to study how they can swap these different metals and what the preferences are towards different metals amongst these different homologs. But we know that these are the 3 modern major homologs today, the molybdenum, vanadium, and iron. So we set the course to understand the ancestral version that the origin of nitrogenase, the geologists inferred is probably around then by means of inferring a presence of nitrogen fixing bugs back in 2.5 billion years ago. But again, we knew the concentration of molybdenum is not consistent. In fact, there's a big shift, coincidentally, coinciding with the great oxidation event. So we started exploring this by using phylogenetics. We basically compiled all the sequences we can find. There are different genes that encode for nitrogenase enzyme. We use the concatenation approach and then predicted the ancestral nodes along the tree, moved on to modeling the structure and then digging through the sequence features, structural features to understand the function. Because we didn't have the experimental system yet, we still want to proceed and understand this computationally first. So moving on to the phylogenetic reconstruction, again, we can dive deep into this, but nitrogenases have different, they're catalyzed by different, it's not a single gene. Basically, there's a gene cluster, I would say, there's the NIF-H, NIF-DK. It's a pretty complex system in a way. And then we managed to reconstruct the tree, we built the tree, and we published this tree in geobiology with the features of the ancestral nitrogenases. So here on the left is the tree. All the blue clades here are molybdenum preferring enzymes. And then on the top is that you will see a different color that all corresponds to different metals. So the orange is iron, and then we have vanadium, and then we have molybdenum, and then we have uncharacterized branches as well along the tree. So here is really the zoomed in version zoomed in clade is where the metal dependency is. So we focused on this, and the ANC A really stands for ancestor A, and then reconstructed the metal ancestors. This makes sense. So that we have the iron vanadium ancestor, ANC B, C, D, we're traveling back in time, and then we focused on this blue node right here to reconstruct the oldest node that connects the both molybdenum and different metal preferring nitrogenases. After we reconstruct the tree, again, your sequence really depends on the quality of your inferred sequences depends on the topology of your first tree. So it's very important that you have quality sequence data, meaning hopefully little gaps as possible. The resource of where you are curating these sequences from also matter. And once you create the tree and it's reliable, and we do this in multiple different ways, we move on to ancestral reconstruction. So here are our the zoomed in part, the top of the tree, if you may recall, where we have different metal preferences. So we focused on basically the residues that we know are important for metal preference. So cysteine or histidine. This was pretty straightforward. Basically, we looked at the literature, we scanned a bunch of papers, and then found out all these experimental studies that assigned certain residues for distinct biochemistry, and then hunted for them in the ancestral versions. Are they there? What does that mean? So on and so forth. So I'm going to skip this part fairly rapidly because this is published. We also have all the sequences available in both our lab GitHub page and also Phylobot, phylogenetic bot that we collaborate with a group in UC San Francisco. It's open source, so anyone can download these sequences and do what you wish with them. Really inspired by the generosities of Rich Lansky. Really, we also want to make this as open as possible, and we would be happy if people used our sequences. We moved on because there were studies in the past that suggested that the cofactor binding pocket volume impacts the metal dependence. So we modeled the metal dependence. We used some machine learning algorithms here. We trained the ancestral sequences, the modern sequences towards different metals, and then implemented that to on ancestral proteins to understand what's the preferred metal binding site, and moved on to sequence analysis. Again, excuse me if I skip this fast, but basically we want to understand the residues on the active site, the cofactor and active site that distinctly discriminate the iron vanadium from molybdenum, and compare the ancestral with the extent, and try to understand what all these ancestors prefer in terms of their metal. So in summary, we found that the oldest ancestors resemble molybdenum more than iron vanadium. So this was interesting because, again, as I said, we knew that the molybdenum in early Earth was low, and then there's a higher availability of molybdenum in the environment. So the fact that the oldest ancestors resembled molybdenum was interesting. That this story is not that straightforward. Just because an element amount is not so high in the environment doesn't mean it's going to be the preferred metal in the system. And of course, you may think, well, there's another possibility that the early nitrogen fixation system maybe wasn't the same as this. And it was a different enzyme or a different ancestor or predecessor than what you see today. So with that, we want to understand what actually is the behind the scene, allowing the biosynthesis of molybdenum into this nitrogen fixing enzyme. So we looked at the cluster of enzymes that actually catalyze allow the active site metal synthesis for nitrogenases. And that's another set of protein as NIF-E and E and N, but also NIF-B, that's a cofactor precursor. So what we found is that in the deepest part of the, we basically want to understand whether these biosynthesis genes are present in the clades that we explored. And what we found is that, well, we have a lot of incomplete data here. We don't know whether these deepest clades have these biosynthesis cluster genes. But it seems that the pathway is incomplete as we go deeper into the tree, meaning that the biosynthesis genes are lacking as we go back in time. Or at a very top level interpretation. So this was interesting. We have molybdenum in the ancestor, but we don't seem to have the pathway that is needed for molybdenum. So that was interesting. That the oldest ancestors did not have a complete biosynthesis pathway. So with that, we try to go a little deeper, that we know that there is no definitive evidence for the deepest part of the clade to fix nitrogen, even though they have nitrogenases. Even though as we come closer to present, there's more and more evidence for nitrogen fixation availability and ability. So the sequence features, even though they suggest that nitrogenase has a molybdenum-dependent ancestry, the oldest ancestors may have lacked a distinct cofactor synthesis pathway that we see today that enables nitrogenase to have molybdenum. So we said, okay, well, either we have an alternative or maybe simplified pathway for the ancestral molybdenum incorporation, or something else is going on here. And then perhaps there may be some unexplored different pathways in current taxa that still depend on molybdenum dependent nitrogenases, but lack a biosynthesis pathway. So this is exciting that there may be different ways to catalyze this system that really is an extremely essential metabolism. So we then looked into an homologue of nitrogenase. So our thinking was straightforward. We said, well, there's another enzyme here. It's called matrase. And matrase is actually helping nitrogenase. You can think of it as like Batman and, no, what was it, Batman? I'm forgetting this. Anyway, but the side guy, you know, the one that doesn't get much attention but helps out. So matrase is that this is the enzyme that participates in the assembly of the nitrogenase active site cofactor, but cannot fix nitrogen. So we dig through the literature. We found these really interesting old papers that suggested that matrases and nitrogenases may actually have an evolutionary root. They function together. They resemble each other so much. They may have come from a single common ancestor. So we set course for this again, and we created a tree that combines nitrogenases and matrases to reconstruct the very ancestor of these two enzymes that function together, and then did the similar analysis for these ancestors, and then look at the sites for the ancestral proteins. So what we found is that the oldest proteins actually resemble matrases, not nitrogenases. So the ancestor of the nitrogenase, and we did this through creating many different trees and many different topologies, and we sort of go as deep as we can really with this, and at some point we have to stop, otherwise we'll go crazy. So because no tree is, I think, ultimately correct. Yes, that's true, but it's so we combined these two genes and reconstructed the ancestor and found that the oldest one is actually does have the features of the extant cofactor biosynthesis protein, so not nitrogenase, meaning that the origin of nitrogenase may not be a molybdenum. May not be actually nitrogenase, even though it's a molybdenum-dependent enzyme. So we may be looking into a completely different pathway of the past, and that may actually still be present here today. We just maybe need to find it or make it in the lab. So while continuing that, as I said, we also engineered the nitrogenase system, and I cannot see the time, so I'm really sorry if, let me check. Oh my God.
You have plenty of time.
Do I? Okay.
Yeah, we're going to 1:30, so.
Oh, okay, all right, I thought maybe I talk for an hour. All right, so so we engineered the ancestral nitrogenase genes inside the modern bacterial genome at the same time. And again, this is for another reason. We want to not only know how these things are functioning, but also connect them to drug record. And it also experimentally evaluates ancestral meta dependence. And really, what's more exciting than understanding the origins of these things? I mean, we study modern life, but just think about the possibility of knowing where the hell these things came from. I think it's just amazing. Anyway, so we have this azotobacter nitrogen fixing bacteria, and then we developed the system. We collaborated with Dennis Steen at Virginia Tech. We were very lucky because I think we were the last visitors that he had to teach this system. So he passed it on, and then we did it. So we inserted we always start with the youngest ancestor inside azotobacteria, and then transform the strains. And this is actually a nice system because we can see if we have a way of assessing whether the nitrogenases are fixing nitrogen just by looking at the plate using ammonium deficient media. So this is much better, and it's a nice system really, that you have a direct selection to understand whether the bugs are fixing nitrogen or not. So and we also we realized that it's very basic, but growth rate assessment to understand just doubling time and the growth properties was lacking for azotobacteria. So we developed this protocol and we published it too. It took us a long time sometimes to assess, obtain protocols, believe it or not. So we wanted to publish all the protocols we ever generate and then make them open source so that others don't have the same challenge. And I think it worked out because we received quite a few emails, especially from grad students, saying that they found the protocol really helpful. So that was nice. But we transformed the strains and they are able to fix nitrogen. This is good news for us. And you're looking at the growth data here. We have the wild type, and then all of these different lines are ancestors. You're looking at 4 different variants. So this is the oldest variant, the one with the straight blue line. So this was a great, exciting day for us. And now we are analyzing their biogeochemical property. But at the same time, we reconstructed them and we also purified these enzymes. And we are studying their function in vitro. We want to understand biochemically what's going on, and we did a lot of library generation to understand whether we can assess the pathway from molybdenum to iron by looking at the amino acids that may be driving this selection. And we are collaborating with Lance Seefeld at Utah State for this, an incredible scientist and collaborator, and very helpful, of course. And Derek Harris is doing the biochemistry work as we speak. So it's pretty exciting time. All right, so I will wrap it up. Ancient nitrogenase is likely preferred molybdenum, and I will end with this this is a big sort of explosion when I talk about this in the astrobiology meetings, because again, we really focus on the geologic dynamics of Earth life and try to understand how that's happening. But really the key is to drill down, not only microbes, but also at the molecular level, understand what's going on and be open to different fields, however long it may take to create these systems, and you know how long it took us. So, but really to go after these things, because I think the protein level innovations is really the key here. If you want to understand the biogeochemical cycles prior to the Precambrian, that's really during the Precambrian, so before the about 500 million years, when the record gets spottier and spottier. But let's face it, where the fun really happened. Whereas the organismal level innovations may be the characteristics for the Phanerozoic, where we can find, perhaps because of that reason, better fossil data. But of course, there are a lot of factors that play into that as well. But enzymes and the ones that go deep may be the key. And I will, end where I started. That we talked about how do we integrate these two records, how do we select these systems, what are the criteria, and how do we integrate and how do we use these two different systems to allow us to collectively understand the past. And even though it seems even it was where we started, the more I think about this, I think the key may be not to really integrate them, but to treat them as a different set of data that prior to combining and prior to creating these stories of this existed in the environment, and then that's why we see this change. perhaps we should decouple these two different records and try to understand them as they are. So try to understand biology, thinking about biology and thinking about what evolution can do, and keeping a liberal mind when we study these things before rushing into making a direct connection with the ancient environment that they lived in. So I think we should resist that urge for it through exploration of what happened at the biologic level. All right. Okay. So with that, I will wrap up. Again, we want to understand ancient life that's on Earth. We use today's biology when we search for life elsewhere. We shall never forget that we are a product of 4 billion year of journey here, and life has been through many things. We are not the end. If we don't, as Sagan said, if we don't kill ourselves and don't kill this planet, you know, there will be more amazing things here. So, and it is happening that we may be very soon confronted with this data that's coming from exoplanets, the planets outside of our solar system, that may suggest that there is an environment available for a nitrogen cycle to potentially emerge. That may mean elements that are available or they match with our understanding of the past on Earth. Should we say that by just looking at the chemical recipe that life may exist here or will? Because that's sort of where we are right now. But we need to think about the past, and I think we need biologists on board to really tackle these questions, because life ultimately is our discipline, and search for life involves what we do. Is very important, and we're gonna get there sooner than we think. So, and of course, oxygen is very important too. These are distinct biosignatures. I didn't talk about that today, but hopefully for another time. Just to finally, we are setting the center also at Madison. It's great. We're going to be a part of the a new origin center that's called YCOR, and then it's going to be launching next year. We're going to be recruiting faculty members, 2 more faculty members interested in early life and origins, but also we will be working together with the astronomers there as well. So that continues. And if you're interested, please email me. We're recruiting a lot of people right now. And with that, I will end. Thank you so much. I want to thank once again to Beacon and for inviting me, for support years. I feel like I grew up here. I feel like an adopted person for Lesky Lab, and it worked out great. So thanks for that. Thanks for being so friendly and supportive over the years. I really appreciate it, and I'm happy to take questions. Thanks.